Hello, my name is Jasmine Johnson. Today is January 29th, 2019, and today I will be doing bacterial morphology and staining techniques for Biology 225. So I actually completed my lab already because the lab was very extensive and I didn't think I would be able to complete the full lab in 4 to 5 minutes. So I'm going to go ahead and go over what I did. So as you can see, I washed my hands prior to gowning up and gloving and putting my mask and my goggles on. So that's what I did as far as safety wise. Also cleaned my area with a 10% bleach solution prior to setting up. I put my paper towel down and I have my alcohol for where my inoculation loops were to be sterilized prior to using them. And here is my E. coli, my S. cerevisiae, and my S. epidermidis. My no longer sterile applicators, but were sterile prior to using. My Sharpie, my lens paper, my staining tray, my crystal violet, my iodine, gram staining, and my saffron gram staining, and my decolorizing gram staining. I also have my candle and my lighter to light my candle. So let me go ahead and explain what we did first. So the first one was a direct mouth swab, and the way I collected that specimen was I grabbed my sterile cotton tip applicator, I swabbed my mouth vigorously, and with a prepared slide, I ended up going ahead and rubbing that area vigorously with the prepared slide. I let that sit and dry. Actually, I grabbed that and I put that over a lit candle so it could asphyxiate. And I did it about 5 times, but not too long, and I let that cool for about 10 minutes. Then from there, I grabbed my crystal violet staining gram stain, and I put about 2 drops in there while it was in my staining container. Let that sit for about 30 seconds to 60 seconds, and then I took that over to my sink and I rinsed the slide with the stain, and I rinsed that until it turned clear. I brought that back to my area so it could dry. And then once that was dry, I went ahead and put that into my microscope, and I looked under each lens of the different strengths, from the scanning to the low to the high, or to the medium to the high. And you will see in my lab report, I went ahead and put a picture of what I've seen underneath the microscope. And the next one I did was an indirect swab, and also with this slide it was called a negative staining that we used, and I used the Congo red staining. So what I did with this one is I also did a mouth swab with the sterile tip applicator, rubbed that over my area. I grabbed my Congo red, went ahead and put a few drops with an additional slide. I dragged that across the slide and then laid it down and let that dry completely. And with that drying completely, after that, I went ahead and I did the same thing with visualizing. So with the, we also have the live specimens from before, like I talked about. And I had my inoculation loops that were inside of my alcohol waiting to be used. And then what I ended up doing to collect the specimen was I got the E. coli first. I went ahead and unscrewed that. I sterilized that over the flame and took my inoculation loop out, put that in, with the prepared slide that says E. coli, I went ahead and I put that over that circle area. Then I also asphyxiated these slides, each one of these, because we wanted each one of the actual specimens to stay asphyxiated to the slide. So with each one of these, I went ahead and I put my staining product in them so that they could wrap the color or decolorize. And I went ahead and rinsed that in my staining tray over a safe area. And once that was dried, I viewed that underneath the microscope, and I visualized what the specimens look like. And then from there, I put the inoculation loops into the 10% bleach. I'm gonna throw this away and clean up the area and let any of my specimens that were contaminated sit into the bleach for 2 hours before actually discarding, and wash my hands and clean this full area. Hopefully I went ahead and explained the procedures of what I did in the lab clearly. Thank you and have a good day.